And I'm going to appear now in a different capacity on the program in my role as director of the RCSB Protein Data Bank, the US Protein Data Bank. And I'm specifically going to talk about the structural biology of the surface of the T cell and the insights that have been gleaned from three-dimensional structures that are in the public domain. And then say something about how this information is impacting our understanding of antibody design for therapeutics, how it's impacted our understanding of T cell function, and then very briefly introduce the concept of the chimeric antigen receptor, not wanting to steal any of the thunder from Novartis, but just to set the stage. And I'll show you how to access the protein data bank, and then acknowledge the very large team effort that's required to make this happen, and say something about the impact that we're having on drug approvals. So it's not an exaggeration to say that the Protein Data Bank was the first open access digital data resource in all of biology. It was established in 1971 with just 7 X-ray crystal structures of proteins, and it's grown over the last 48 plus years to be the single global archive for all experimental 3D structure data for proteins, DNA, and RNA that's put into the public domain. And today there are now in excess, I think, of 157,000 structures in the PDB archive. Instead of the archive being managed solely by the United States at the expense of the US taxpayer, we now jointly manage the archive with collaborators in Europe, PDB Europe, and Asia, PDB Japan, and we also work closely with another data resource for structural biology, the so-called Biomag ResBank for NMR data. And 4 of the structures that inspired the launch of the PDB and the commitment to open access before it was even a thing are shown here on the right. And they include myoglobin, the very first structure to be determined by Sir John Kendrew, followed closely by hemoglobin, the work of Max Perutz. And then 2 of the 3 early enzyme structures are shown here, lysozyme and ribonuclease. I did my PhD, D.Phil, in the lab of Sir David Phillips, later Lord Phillips, who determined the structure of lysozyme and published the first enzyme structure. We started the PDB with very, very small protein structures. This is drawn to scale. So some of the pancreatic enzymes, etc., myoglobin, hemoglobin, are depicted here. And over the last nearly 5 decades, the wealth of structure information that has made its way into the archive and the diversity of the kinds of 3D structures that are available is depicted here. So we've now got a large number of integral membrane protein structures in the PDB. We have virus structures in the PDB. We understand muscle locomotion at the molecular level. We understand DNA. We understand DNA replication, transcription, translation, a whole host of phenomena which were previously just sort of boxes in the middle of cartoons. We've got very detailed 3D structure information, and the importance of this is that knowledge of the structure tells you how the molecules work. It can explain what happens when things go wrong, when there are non-synonymous mutations, as you've seen from some of the work that Ned showed. And it provides a picture of biology that goes well beyond the names and arrows on metabolic pathways, etc. So this organization, the PDB, has been in existence for 48 years. The RCSB Protein Data Bank has been in existence now for more than 20 years. It moved to Rutgers from the Brookhaven National Laboratory on Long Island in the late 90s, and now includes teams at UCSD and UCSF. We collaborate, as I said, with our Worldwide Protein Data Bank partners. We have 2 websites, rcsb.org for researchers and for outreach and education, the PDB 101, the introductory course. We're supported jointly by NSF, NIH, and DOE. And here's a very happy team when we crossed the 150,000th structure just a few weeks ago. These structures touch on fundamental biology, on biomedicine and energy, and explaining why we have funding from NSF, NIH, and the Department of Energy. And our job is to turn global data into global knowledge. There are approximately 31,000 structural biologists, people who have deposited data to the Protein Data Bank over the years. And this data comes in on a weekly basis. There's a new release of the archive every Wednesday. And those data are immediately sucked up by more than 400 distinct scientific data resources worldwide. They are injected into our website, where millions of unique users per year come to learn about proteins and biology, and into our educational website, where more than 600,000 users per year come in to the system. So let me talk very briefly about the impact on antibodies. The very first human structure that came into the Protein Data Bank was that of an IgG light chain dimer. It was purified, a so-called Bence-Jones protein, purified from the urine of a patient with multiple myeloma. The first FAB fragment came into the archive in the mid-80s, and the first single chain FV structure came into the archive in 1994. Since these early structures, literally thousands of antibody and FAB structures have been deposited into the PDB. And this information is used on a daily basis by individuals working in biopharmaceutical companies who are developing new biologic agents. And our friend Gary Gilliland, who was involved with the RCSB PDB many years ago, is quoted in this article that he published in a book in 2012. Engineering of therapeutic antibodies is based to a large extent on our knowledge of antibody structure. And you can see this very clearly in the design of the single chain FV depicted here. This is Protein Data Bank ID 2 Kilo Hotel 2 using the NATO alphabet. You can see the linker between the variable regions of the light and heavy chains, recognizing this cytokine IL-1 beta. So what we're here to talk about today is really to understand how it is that the T cell is seeing antigens and able to respond to these antigens. So this slide, or this image I got from Steve Almer, who's the next speaker, showing the immune synapse and the T cell interacting with the antigen presenting cell. As Ned described, there's a molecule, or it's actually a complex of two proteins that sits on the surface of this cell, on this cell, that presents linear peptide antigens to the T cell receptor. And I don't think it's an exaggeration to say that until this structure was deposited to the Protein Data Bank back in 1987 from the lab of the late Don Wiley, work of Pam Bjorkman, who's at Caltech now, PDB 1 Hotel Lima Alpha. Until we saw this structure, we didn't understand how it was that linear peptide antigens were presented by cells to the immune system. And since this publication in Nature, more than 750 structures have come into the PDB, which together help explain how T cells work. So the next important milestone was this, the visualization of a linear peptide antigen in that groove in the act of being presented to the T cell. And subsequently, we saw structures like this, in which there was recognition of the MHC antigen complex by the T cell receptor. And it was these observations that made sense of a lot of black box immunology that was it's not an exaggeration to say it was a mystery at the time. Our understanding has developed enormously since those early days. And we now realize that the T cell receptor and the interaction with the MHC complex with the peptide is just part of the story. And there's a series of additional signals which can be received by the T cell. Some stimulate the response of the T cell, as you heard from Ned. Others inhibit the response of the T cell, and he's already mentioned CTLA4 and PD1. I'll go into a little bit more detail about those in a moment. So the interaction between CTLA4 and B7-1 serves to downregulate the response of the T cell when an antigen is presented. So if you have a tumor cell that has this protein on its surface and a non-self antigen is presented to the T cell receptor, it's quite likely that the T cell will say, okay, I recognize that this is not a self antigen, but I'm not going to kill the cell that's presenting it because I'm receiving this downregulatory signal as a result of the interaction of B7-1 with CTLA4 sitting on the surface of the T cell. And there's a host of structures of CTLA4 components, individual domains typically in the PDB dating back from 2000. There are also a handful of structures of B7-1 and B7-2 in the PDB, again dating from around 2000, 2001. And one of these structures shown here actually captures the interaction of the B7-1 dimer with two molecules of CTLA4. The T cell would be up here, the antigen presenting cell would be up here, and to the left would be the T cell, would be the MHC interacting with the T cell receptor. And it's this molecular interaction that's responsible for the downregulatory events that occur that help the tumor survive, give the tumor a competitive advantage because it's expressing this protein on its surface. We can interdict that with an anti-CTLA4 antibody called ipilimumab, otherwise known as Yervoy, and there's a structure in the PDB of which was not made public until after this drug was approved, the biologic was approved. So here's the FAB light and heavy chains interacting with CTLA4. This work came from Steve Almo's lab. So this is the drug in action, recognizing CTLA4 and precluding the possibility of CTLA4 being engaged with B7-1. Similarly, we understand in 3D the events that occur at between the antigen presenting cell and the T cell when PDL1 or PDL2 interact with PD1. So this again is the T cell. The antigen presenting cell is down here, and there are more than 30 structures in the PDB, many of which have actually been determined and contributed by Steve Almo's lab at Albert Einstein College of Medicine that reveal, exactly how PDL1 and or PDL2 engage PD1 and downregulate the T cell. And we now have 3 US FDA approved anti-PD1 antibodies available for treatment. The first 2 that were both approved in 2014, pembrolizumab from Merck, nivolumab from BMS. I should have mentioned that Yervoy came from BMS. That was a very significant pioneer in this area. So here's the interaction of PDL1 with PD1. Here's the interaction of PDL2 with PD1. And we have structures of the FAB fragment of pembrolizumab interacting with PD1 and nivolumab interacting with PD1. And you can see one of the things that's interesting about the comparison of these 2 views is you've got 2 scaffolds here that are very, very similar, but the precise mode of interaction with the antigen, with the target protein is actually different comparing the 2. So they are interacting with different portions of the surface of the protein. And information like this, where we can map the binding site of the antibody onto the target protein, is sometimes used by pharmaceutical companies to try and strengthen intellectual property claims when it comes to biologics, because you can't get a composition of matter patent on this protein. You can't actually control the manufacture well enough to get a composition of matter patent. So one of the things that's done is to try to claim a sort of mechanism of action with respect to the part of the molecular surface of the target that's being occluded. This hasn't been adjudicated in the courts yet, but there is a case making its way through the system, I'm told, that will potentially produce a ruling on whether or not this kind of IP claim is relevant or not. If PD1 is a target for a monoclonal antibody that blocks the interaction with PDL1 or PDL2, it stands to reason the same must be true of PDL1, and that's exactly what a number of companies have shown. So since between 2010 and 2018, 3 monoclonal antibodies for treatment of cancer were approved, that all of which bind to PDL1. So they're now binding to the protein that's on the surface of the tumor cell. And you can see again, they're interacting with different parts of the 3-dimensional structure of this apical domain of PDL1 as it extends from the surface of the antigen presenting cell, the tumor cell. It's also been shown by work, again, this is at BMS, that a small molecule can be used to actually blockade the PD1, PDL1 interaction. And the strategy here was not to interfere, not to try to break the PD1, PDL1 interaction, to break that, open that large protein-protein interface. The energetics of doing that's actually very challenging. The strategy, the mechanism of action strategy that was pursued by the team at BMS was try to identify small molecules that cause dimerization of PDL1 on the surface of the tumor cell such that it couldn't interact with PD1 because the part of the molecular surface that's necessary for the interaction of PD1 was occluded. And that's what you see in this rendition here. You can see the small molecule sitting in the middle of the dimer interface between the 2 copies of the apical domain of PDL1, and thereby precluding the possibility of engagement with PD1. This is an investigational agent. I don't think it's even been put into man, but it's intriguing to think that that a small molecule strategy could be used to accomplish similar ends. I say this because if we're going to democratize access to immune checkpoint therapies, it has to involve small molecules ultimately. We're not going to see large numbers of patients treated outside the West and Japan, Korea, unless with immune checkpoint strategies, unless it can be done with a small molecule. The cost of biologics is simply going to be too great in those geographies. So in addition to this strategy, the other obvious thing to do is to think about what happens inside the T cell after the engagement occurs between PD1 and PDL1, and go after the cell signaling, the intracellular cell signaling targets that would permit blockade of the consequences of the interaction with a small molecule. So let me now introduce the CAR T cell concept. So we talked about the chimeric antigen receptor. So you know a chimera in mythology was an animal, a beast that was created of there's parts of 2 separate animals. So a lion and a dog, for example, would be a chimera. So in this case, the chimera consists of parts of different proteins. And in the simplest incarnation, the notion was that you have a molecule sitting on the surface of the engineered T cell that's capable of recognizing an antigen on the target cell. And the preferred vehicle here is actually a single chain FV fragment. I showed you those earlier, and those would occur as a pair. There would be a transcellular portion, a linker segment, that crosses the cell membrane and induces the dimerization here. And then on the inside of the engineered T cell, you're going to have sequences that are capable of recruiting signaling proteins that activate the T cell. And this particular example depicted here, it's a sequence that's recognized by ZAP70, which is a known activator of cell killing. This particular model of the chimera was a consequence of assembling 4 different structures from the parts of 4 different structures from the PDB in postulating what this particular chimeric antigen receptor would look like in the cell interacting with ZAP70. If you go to our PDB 101 website, pdb101.rcsb.org, and look at molecule of the month and simply search with CAR T, you'll see David Goodsell's article published from 2017, which explains the molecular anatomy of this simplified chimeric antigen receptor. The one that's actually been approved, that's at work in the clinic today, transforming the lives of patients and their families, is a little bit more complicated than that. And I think you'll hear about this more from Randy Isaacs when she presents on behalf of Novartis. So let me close by talking about the way in which to access the PDB data for you researchers. The principal route will be through rcsb.org. Here's a picture of our website. I took this just on Sunday, so it's current. You can see the latest molecule of the month article concerned ribonucleotide reductase. It was actually co-authored by one of our summer interns who worked with David Goodsell on putting this out. Out in the foyer, there are some calendars celebrating the 20th anniversary of Molecule of the Month. David has literally written close to 240 articles for this over the last 20 years, and the 240th will appear in December of this year. Most of them he's written by himself, but occasionally he's written in collaboration with students and members of staff with very good effect. So there are a whole host of different ways to search. You can put in the amino acid sequence of the protein you're looking for here. You can put in the name of the protein. You can put in the PDB ID, and when you do any one of these things, you'll come up with a list of hits. If you put the PDB ID in, so this is 5 Juliet X-ray Echo. This is one of the structures of the FAB bound to PD1. So when 5 Juliet X-ray Echo comes up, you can see the title of the entry here, the PDB entry. So human PD1 ectodomain complex with the pembrolizumab FAB. And then in this graphical window, you can immediately see, okay, here's the FAB fragment, here's PD1, and it's possible to explore this in a lot more detail in 3D. Also provided on this page, but not visible, there's information about the publication that resulted from the structure. Close to 90% of the structures in the PDB have been published in peer-reviewed papers. You'll also see information about structure quality, and if one needs to get access to the underlying experimental information, that's also available a little bit further down the page. You can also identify all other proteins in the PDB that are similar in sequence or similar in structure. There's a host of biochemical and biologic annotations that are also available to you. These are updated on a weekly basis. So this particular PDB entry, this structure summary page, actually represents essentially an updated version of the original publication. That's frozen in time. It may well be that a biological function that is discovered subsequent to the structure coming into the PDB and the structure being published, that information can be found on the annotation tab. So this is a living digital data resource that is literally being used by millions of people around the world. In 2018, we recorded nearly 750 million structured data file downloads from the archive. So that's more than 2 million per day around the world. So I've told you about the origins and the history of the PDB. I've shown you what we're doing to add value to the atomic coordinates that are available. I said something about our impact on understanding of antibodies, T-cell function, introduced the chimeric antigen receptor to you, and given you a teaser as to how to get access. After I acknowledged the contributions of the very large team of people that are necessary for making this all work, I want to show you some additional data looking at the impact of the PDB on drug approvals. Just a single slide. So this is the rogues gallery. These are all the individuals who are currently on a full-time basis contributing to the smooth operation of the protein data bank. David Goodsell is depicted here. He's actually based out at Scripps in San Diego. Shown here is Helen Berman, who was responsible in the late 90s for literally saving the PDB from collapsing under its own weight. She brought it here following a recompetition from Brookhaven National Lab. The NSF, NIH, and DOE shifted the funding here to Rutgers, and she took what were a bunch of flat files and turned them into the relational database that is now underpinning everything that you've seen. At that time, there were fewer than 10,000 structures in the PDB. So we're now 15 times larger. And it's not an exaggeration to say that had it stayed at Brookhaven under the previous management, it would literally have collapsed. The newest entrant to the PDB team is Andre Saleh, who's based at UCSF. He's an expert in so-called integrative structure determination. This is the approach to structural biology that uses not one method, not X-ray crystallography alone or electron microscopy alone, but a combination of methods to tackle much larger, more complex structures that are going to be, in many cases, the drug discovery targets of the future. I say that this is important for drug discovery because of work that we were able to publish a little bit earlier this year. This is principally the work of John Westbrook, the senior most programmer and architect of the PDB, working here at Rutgers. So we looked at the 210 new drugs that were approved by US FDA 2010 through 2016. And we asked a very simple question, which is, what fraction of those actually would have been impacted by PDB data? And what we're able to show is that 184 of these 210 new drug approvals had their target structures in the PDB, most of them more than 10 years before the drug was approved. And then we looked in more detail for examples of where structure-guided drug discovery was used to actually refine the fit between the small molecule and the target protein. And an example of that is shown here with vemurafenib bound to the V600E mutant form of BRAF kinase that's found in 50% of patients with late-stage melanomas. And prior to the appearance of the immune checkpoint therapies, this was the go-to medication for patients with melanoma. It extended life expectancy, but unfortunately, because it's a targeted therapy, because it was used as a single agent, of course, resistance begins to emerge fairly rapidly. So the standard of care now, if you're going to receive a BRAF kinase inhibitor, is that it's co-administered with a MEK inhibitor, and Novartis in 2018 had an approval of encorafenib in collaboration with, I think, in combination, I think it was binimetinib, is that right? Yeah, and that's now the standard of care for targeted therapy for melanoma. The results seem to be superior at the moment for patients who receive as first line receive the checkpoint therapies, but I think we're still the clinicians are still trying to learn how to use these in combination, and I think we can anticipate that there will be further improvements in survival for melanoma patients. We just completed, still unpublished, an analysis of US FDA oncologic drugs approved 2010 through 2018. And I'm pleased to report that in 70% of the cases for the small molecule approvals, those were the product of structure-based drug discovery done at companies. So the kind of work that was done at Plexicon for vemurafenib is the rule, not the exception in terms of drugs. And it demonstrates the importance of PDB data, because of course those were the structures that are in the public domain are the starting points for what the companies do when they begin a structure-guided drug discovery campaign. These are of importance in drug discovery that go well beyond basic science. These are making direct contributions to the likelihood of success of drug discovery programs in biopharmaceutical companies around the world. Every major company's got a big macromolecular crystallography team, got a big computational chemistry team, and they're using this kind of information on a daily basis to produce new drugs.